So today I want to talk about the origin of life, what we know about it, what we don't know about it, what people claim we know about it, and what the evidence looks like, and in particular relating to the timing. So the question that we'll try to get to at the end is if we think that there was life, 4 billion years ago on this planet or not, sort of what form does that evidence look like? Because the further you go back, the worse shape the geologic record is in, the less rocks you have, the less material you generally have to work with. So first, as a disclaimer, I'm going to be a bit of a wet blanket at first. This is a topic that attracts an enormous amount of attention because if you find extraterrestrial life, you're guaranteed to be famous. And therefore, people try and put a lot out there. And so, last time somebody raised ALH 84001, where there was a nice press conference in the 90s. And, well, I'll summarize my feelings here. This is basically bullshit. This is a weathered meteorite that carbonates formed in. And so, people got really enthusiastic, and they said, oh, look, here's something, this could be biogenic, and that was essentially claimed. I mean, you'll find secondary carbonates that look like this on Earth that have nothing to do with biology whatsoever. There's even, this gets worse, people claim that they find Satyr Bacteria in carbonaceous meteorites. For example, they like finding this in a meteorite called Orgueil. Orgueil's been sitting in the drawer of a French museum for about 100 years. So the odds that if you don't find life in there, I'd be heavily surprised because it's been stored in an incredibly dirty environment. But also the arguments are basically the same. You see something and say, oh, this looks like it could be a bacteria, and then you end up writing papers. You've heard, so this tends to get published as a journal of Cosmology, which is a legitimate enough sounding journal, even claims it's peer-reviewed. It isn't, it's none of these things. This is basically a crank website where people publish a surprising number of claims of extraterrestrial life end up appearing here. This is not a legitimate journal despite its name. So this is just to sort of go through the, there's a lot of attention, and basically sort of the text that I'll just summarize is, if you're wrong on this topic, the costs to you as a person aren't high. If you happen to be correct, you will be one of the most famous people that's ever existed on this planet, right? People will say your name in the same breath as Einstein and Newton for a time immemorial because you've discovered extraterrestrial life. So I'll go with Carl Sagan, extraordinary claims, extraordinary evidence, and most of these things end up being sort of qualitative arguments based upon the shapes of some carbonates that are ultimately do not rise up to the level of extraordinary claims, or extraordinary evidence, sorry, for their claims. So basically, this is like doing and publishing this type of stuff is like playing the lottery. If you happen to be right, you've won. So a lot of substandard work gets attention, and well. So let's get into some of the, let's skip ahead to some of the things that might be a little more concrete and more important. So the place I want to start out with is some of the things that are required, some of the building blocks, and maybe what we know about how we made them. So then we can ultimately go on and look at the geologic record and try to see if there's what the evidence for early life is. So one of the key things that you need is you need amino acids. These are the building blocks of proteins, of DNA. This is one of the most important things. You need carbon, hydrogen, oxygen, nitrogen. You need phosphorus. You need some really cool elements. You need a way to make them. For example, humans only make some of the amino acids that we need. We can't actually make all of them. Some of them have to come from the food that we need. So we need, say for human life, we need a way to make amino acids that are outside of our body. So can we make them abiotically? The most famous among this is the Urey and Miller experiments. There's Harold Urey. These were done at the University of Chicago, who basically looked at something like a Jupiter atmosphere. It was a reducing atmosphere, and said, what happens if we have lightning strikes happening in this atmosphere? So they set up a really neat experiment where they put in, they had a capsule here. They had water, methane, ammonia, hydrogen and carbon monoxide. They put the gases in, they cooled it, and then they basically put an electric spark through it to see what would happen with the chemistry. So basically, can you combust this stuff and make something interesting? And the answer to this is yes. They actually, in the original experiment, found 5 amino acids so that you can clearly make some complicated molecules out of a very simple mixture. So it's possible then that you can make, aspartic acid, glycine, and these other ones in an early atmosphere that is composed of this composition. There are some pictures of what these apparatuses might look like. There's your glass bulb, for example. So this seemed like a very promising way, except that the record for the early Earth, as I showed you last week, sort of disagrees with these conditions. The odds are that the early atmosphere was not reducing. That was more oxidizing, so that instead of methane you had, or carbon monoxide, you had CO2. So that you'd need an entirely different, and unfortunately a lot less reactive gas mixture. Methane, natural gas, is a very reactive molecule. CO2 is a very stable molecule, so that actually makes the gas chemistry a lot harder to do. So this kind of pulls the plug on Miller-Urey being a nice viable mechanism, at least on Earth, but maybe you made these elsewhere and transported them here. So people do find amino acids in meteorites. Here's just a nice table since I couldn't figure out what a good figure is, so don't worry too much. But here's a list of amino acids, and then they find them in some contribution. So this is parts per billion. So basically, there's 100 parts per billion. So there's very little, but there's some amount of amino acids in meteorites. As far as how you make them, the mechanism isn't quite agreed upon, but there's a really cool one that was published. This is work from Fred Chesla, who is a professor in the Geophysical Sciences Department, and posited the following question. If you have ice grains as you're forming your early solar system, so this here is a snapshot of a model where you have the protoplanetary disk. So while the planets are accretion, after the gas has fallen down into sort of a disk from the molecular tab, this is the flux of UV photons from the early active sun. And you can see it's really high out here, really high down here, and kind of low in the midplane. This is because this is optically dense. There's a lot of dust and gas and other stuff that's absorbing the UV, and so you don't tend to get it. And what he showed is that actually, if you track ice grains, they have this white line here is one of these crazy paths that ice grains can take through this early protoplanetary disk. And occasionally, what they noticed is that occasionally you'll take excursions where they're getting UV, but then they come back in to the shielded part. So basically, they're seeing UV photons, they're getting heated, so you can do some chemistry in lab settings. We know that reacting sort of primitive carbon species, especially in ices with UV photons, can make very complex organic molecules. So basically, what the model is positing is that even though it spends very little time here, most of the time it's cold, so you're not going to melt and destroy it, but you can get quite a few, 10 to the 10, 10 to the 12 photons over the first sort of million years, so while this process is happening. And what this model is really arguing, and this is sort of, so radius, so 1 AU, there's the Earth orbit, so this is happening. You're getting particles that see these high fluxes, right? This is an enormous scale. The inner solar system is kind of here. So in the outer solar system where you have these ices, you can produce, you can use the UV's photon flux to make some organic materials, and then this might be a common artifact of planet formation. So that because you have UV interactions, you have these gases, they have some carbon-bearing species in them, that you can make some cool complex molecules that can then get incorporated into the meteorite record. They can get delivered to the early Earth. So this seems like one way to maybe get some materials here. You also have around stars. So here is, if you look right here, you can see this band. This is a feature observed in spectroscopy. This is a wavelength of about, this is 957.7 nanometers. This is evidence for C60 plus fullerenes or buckyballs, and so you have really complex things, but if you, this is from a review paper, there's evidence that chemical pathways similar to combustion processes on Earth. So this is basically when you take things like methane and other molecules and you burn them, you make soot, and that soot has large carbon molecules in it, including buckyball. So this is one way to make them sort of in outer space, and that could be so that you can make molecular synthesis in circumstellar environments on timescales as short as several hundred years. So this might be another way to make some material that might get incorporated and be of use, but is not necessarily evidence for, say, extraterrestrial life every time you look at a star. So there's abiotic ways to make this stuff that we need as these ingredients, the amino acids and these sort of heavy carbon molecules are really useful for life as we know it. The other way to sort of do this in outer space is that as you have sort of dust grains, they will attract little carbon molecules or maybe some H2O, some methane, and over time these might find each other and slowly react because they're adhering to the surface of the dust. So you have these simple carbon molecules happening, and if they get a little bit of energy, they're next to each other as they're all stuck onto this dust, because remember the interstellar space is really cold, so stuff tends to want to stick. You can make things here. In this case, they're making a sugar molecule on dust, and this was prompted because it was actually observed spectroscopically. So there's a whole bunch of really cool extraterrestrial chemistry that is happening abiotically, but it's not clear how to relate that back to the life conditions. And it's in some sense there's that organic is to the chemists anything that involves carbon and such, whereas to people when you talk it sort of means life. So we really should be using the word biogenic, and this is abiogenic, so without the involvement of life. So, unfortunately, there's not an awful lot of agreement on the origin of life because we only have one example, right? This is the hard part of studying it. We only have life on Earth as we know it today. We certainly don't have the environment where it came from. So there's some hypotheses that have been presented. I'll show you my favorite on the next slide, but these are all in the running. So we really have no idea how this actually happened on Earth. People have hypothesized. One of the ideas is, for example, that before you had DNA, which is the double helix, that you only had RNA. This is the RNA world hypothesis. So these are the things like in viruses, which only have RNA, that this precedes and then came DNA, but it doesn't answer the question, how did we actually end up with the life, the RNA-based life. So there's possible settings where this could have originated. There's deep sea vents. These are really cool because you have an enormous chemical gradient. You have heat and gases coming from the mantle being put in contact with liquid water, so that immediately gives you huge chemical gradients and might be a great energy source for early life. You have arguments that it might have happened in hydrothermal fields. So near volcanoes, you have a lot of hot water. This is where you sort of have hot springs, and that these hot springs might be a great place to make life. I'll talk about this one on the next slide. People have argued that muscovite, which is a sort of platy mineral that is actually used for insulation quite a bit, that the different layers provided protection and were able to assemble life in there. You could make it in clays, maybe you get it from somewhere else, panspermia, and then there's many more. I could fill up the rest of this list, and the problem is no observational evidence from the geologic record favors any one of these ideas over the other, because the record, as we'll get into, is just not good enough to do this. So that's the unfortunately disappointing part of the lecture. The part that I do like is I really like this hydrothermal idea. So this idea came from the observation that cells contain a lot of potassium, in particular to the amount of sodium that they have. And the idea is that early cells were not able to, did not have membranes that could keep ions out, so that they were basically much more, so right now each cell has a membrane on it that is able to keep ions out and limit that exchange across. And the argument is that early life was not able to do this, and that if you look at the fluid inside cells, it's actually massively out of equilibrium with what you you say for a seawater composition. So basically, if you have an early cell that is not able to select ions, it should be massively enriched in sodium, but not necessarily potassium. But what we actually see is that it's massively enriched in potassium over sodium. And so this argument, this was made by this was made by a bunch of biologists. So they went and looked at this, and they argued that this composition, that the fluids in the cells are recording these early conditions that would have been in equilibrium with the cell, and that these were heavy in things like potassium, phosphorus, transition metals, but particularly it's the potassium to sodium ratio that's really challenging for seawater. Seawater's got a lot of sodium and not a lot of potassium. And they argue that what could happen is you have a magma chamber, you have water infiltrating it, and that you could end up making environments where you separate sodium from potassium by having vapor, and then you can have, ammonia, CO2, so you can have a lot of the cool stuff that you need, and you have a lot of potassium over the sodium, because the sodium is going to stay in the water, so as the water gets hot, it is going to come out and might cool as the water, whereas in other places it might actually come out as vapor, and the vapor is going to be enriched with potassium. The part about this idea that I have found it really interesting is that if you look at the rock record, so these are the black are rocks between 0 and 1 billion years, 1 and 2, 2 and 3, 3 and 4 billion years, so we don't have a rock record in the Hadean, so this cuts off before the interesting time period. But what I want you to notice is, right, we've talked a lot about there being evidence for high silica content early in the crust, that this is SiO2, so your granites are going to be up here, your basalt, your sort of Hawaii is down over here, and that if you actually look, since we have evidence for felsic crust, this is the sodium to potassium ratio. So in mafic rocks, in an early mafic crust, you would expect a lot, really high, so 4 times as much sodium as potassium. And if you go to a much more felsic crust, you actually would expect a much lower sodium to potassium ratio. That is to say, you expect a lot more potassium. So if you have this process taking place, since the early continents looked like they had a lot of high silica rocks on it, I think this is an entirely cool observation, that they're requiring a high potassium sodium environment, and on the other hand, the geologic record is telling us that we had a high potassium sodium part, that we had this crust that was high in, that was seemingly compatible with this process. So I think that's really cool that you can come at this from 2 different directions and sort of find a little bit of agreement. Doesn't prove that life happened there, but I think this is a really cool idea and has become my personal favorite, is that what happened is you had a hot spring on the early continental crust, and that in those hot springs and those hydrothermal systems is where life originated, because you can bring in stuff from the surface into the lake, you can feed it with things that are reactive, you can get that potassium. This process, the leaching is also much easier, so this liquid would be really high in potassium and low in sodium. So I just think that this is probably my pet favorite, the one that is most commonly accepted. The one that's probably the most popular is the origin of deep sea vents. That there was that in these black smokers you had is where the origin of life. And that's probably the most accepted one. This here is my personal pet favorite, so it comes with. Okay.
I may be missing something, but if you're saying that the reason that it's postulated that the potassium level is so high compared to sodium, because it was in equilibrium with the environment that existed when, during early times, over 500 million years ago. The cells that you're measuring weren't made then; they were made recently.
The argument is that this is evolutionary, really hard to iron out. Sorry, I skipped that part. So that evolutionary, this is something that's not going to change. So people who do like the molecular clockwork and look at like what genes have changed and what part of the expression has changed, that this is some of the base scaffolding of how cells are made, and is among the slowest things to change. So these are the features of the cell that have seen the least amount of biologic activity, that have seen the least changes. So you're measuring this in cells today, that is correct, but the argument is that this is something that doesn't actually change all that much, because it's this sort of base infrastructure of the cell.
Has to do with the chemistry of the cells and the way they operate?
yes, it's that over time, evolution has happened, has changed a lot of features of the cell, but that there are certain parts that are less likely to change than others. And the argument is, if you can, in their paper, which is gets into what parts of the genome of cell is, how easily they are to change. The parts that determine the potassium and sodium, like the fluid composition, are the hardest parts of the genetic code for the cells to change. So this is the stuff that is likely recording back through, and evolution hasn't really done that much to it.
And so the modern composition of cells, the modern ratio of potassium, reflects the earliest origins of the first living things. They had that composition because that was the composition of the water around them.
That is the argument, yes.
They had to have that ratio of potassium and phosphorus because they did not have cell membranes to control transmission. That cell membranes that were able to control this are a later feature of cells, and that the earliest cells may not have possessed this. But modern biochemistry reflects the earliest origins of cells. That's why it's preserved.
Yes, that is the argument.
Okay.
Yeah.
The graph on the right hand is from rocks or from the smoker vents?
This is from rocks. So these are rocks, but this is what's being leached. So if you look at their step, right? Meteoric water gets heated by a magma chamber and then leaches the materials. And the point is that in a high silica crust, the starting material that you're leaching from is already much more favorable to where you need to get. Right? If you have 3 times as much sodium as potassium in the rock, you need a really efficient process that gets potassium out over sodium. But if you have a starting material with, maybe equal or a little bit more sodium than potassium, your process doesn't have to actually be all that efficient. So it's just easier to do it. That's the point of the figure on the right, is that this is sort of the starting material that the water is going through and bleaching the materials from, that you can then ultimately make your hydrothermal vent at the surface. So basically, it refers to the composition of the rocks that are getting leached by the fluid. The point is, we have evidence for these, and if you were to leach stuff from them, it's a lot easier to get to these high potassium sodium environments.
The hydrothermal vents would have been underground then?
At the surface. Think hot springs.
Okay, but one of the arguments for deep sea vents is that they'd be far enough underwater that they would not be affected by cosmic rays and by high energy ultraviolet light.
So the penetration of high energy ultraviolet light would depend upon the atmosphere composition. We don't know all that much about the thickness of the atmosphere, so this may not be a problem that you need to solve necessarily. You also don't the penetration depth isn't necessarily all that deep. So if you have a lake, right, if you go swimming, by the time you get, if you have ever been, gone underwater and opened your eyes, it gets pretty dark pretty quickly. So you don't need necessarily all that much water to protect yourself from the interactions from outer space.
So hot springs on the surface, 100 feet down perhaps?
Probably not even 100 feet down, probably several meters, maybe. The other difficulty, the most difficult part of studying the origin of life itself, is that life, annoyingly enough, is everywhere. Hot springs, here are the Yellowstone hot springs, massively contaminated. Life is everywhere in here. The Atacama Desert is the driest place on Earth. This is in Chile. And people like JPL, when they try to test their instrumentation, this is a hard place to find life, but you can do it. So, like teams from JPL, when they're trying to develop methods for detecting life, they go out here and see if they can find life here, and they tend to. You can even find it in subduction zones. So there's sort of deep life that's underground, and then here is, depth, and you have basically the upper limit, the hottest thing in which we found life is 120 degrees C, and you can basically draw this line here, and you have opening and cracks, and so you can get life down pretty deep into the crust. There's sort of models that this can, that life is, the point is that life has gotten everywhere, and that we've even probably subducted a huge amount of organic carbon and contaminated the mantle coming out. So it's not clear, we're going to get to carbon isotopes, what would happen on a planet without life. So this later contamination issue is pervasive and makes this a really difficult thing to study. So how do we determine when life began? This is the sort of, we'll get into the geologic record. We'll just try to figure out when do we have the origin of life, or at least the evidence. So here's a time scale. There's Nixon resigning, basically at 0. The extinction of the dinosaurs. This is 65 million years ago. 500 million years ago. This is the Cambrian explosion. This is when basically this is when you got this is when you have most of the big fossils happening in rocks that are 540 million years younger. The oldest eukaryotes, 1500. Oxygen accumulated in the atmosphere at about 2.3 billion years ago, so this is what we all breathe. The eukaryotes, the oldest fossil here will show what that looks like, is about 3.4 billion years old, though this is a heavily debated, disputed thing. And then we'll get into the isotopic evidence. So the point is, most of the stuff, like from fossils and such, is pretty recent. So the macroscopic fossils are really only showing us about life in the last sort of 15% of the age of the planet, and it's this entire period, known as the Precambrian, labeled here as the Precambrian and the Hadean, where it's really difficult to study life because we tend not to find large fossils. So here's an example. This is a picture of a fossil that is about almost 3.5 billion years old, or at least it's claimed that this is a fossil. We do have some organic materials, some organic kerogens that go back to about 3.5 billion years. And what you will notice is that we're going to talk about carbon isotopes. So this again, this is the amount of 13C divided by 12C relative to a standard material. And basically, the bulk Earth for this scale is at 0 carbonate. So this is, and organic materials are at minus 25 parts per thousand. So they are basically 2.5% depleted in carbon-13. And this is because life is cheap energetically. It prefers carbon-12. We know this from, organic kerogens at present day have this offset going back basically to 3.5 billion years ago. And life just really likes carbon-12. It's energetically, it's a lot easier to move around because you only have to move 12 out of 13 of the mass. So you don't have to move all the mass of a 13C. And that this offset between carbonates that basically not life involved versus the kerogens, which are sort of life involved, has been a constant offset for 3.5 billion years. So this is pretty good evidence of life going back at least this far. So here's some more images up here. These are some of the claimed sort of oldest fossils. They're found in the Strelley Pool Formation. These are 3.43 billion years old. And in Western Australia, if you're following along, the Jack Hills are somewhere like here on this plot, where all the old Zircons are from. So Australia is a great place to get old stuff from. It's been pretty geologically stable. So here's an example of an old fossil. This you can take this as any amount of how convinced you are. I won't try and sway your influence or influence you on that. I personally never found this to be super compelling. There are stromatolites. These are sort of bacterial growth, where there's a bacterial colony that is growing, and then it takes in sediments. And so you end up, like this is Shark Bay in Western Australia, this is a modern one. In modern ones, they're biological, so you can sort of cut these things open, look at what they look like, and people then go, here is an example. This is from Greenland, the stromatolites. Where they found them, they claim that they are sort of 3.8 billion years old, and they have this bacterial community, and their evidence basically amounts to drawing in some squiggly lines, so that's not super convincing either. The bigger problem is, in a review paper here by Tanya Bosik at MIT, who spends a lot of time trying to understand and model stromatolites, is that there are ways to get things that look an awful lot like stromatolites that don't involve any life whatsoever. And so the testing of whether or not these old stromatolites that people find, these old, what they're claiming are bacterial colonies, are basically along arguments that they have similar. So here's the old one that they're claiming, and the younger one that they're saying analog. They're saying this looks awfully similar, but that's the level of the argument. And people who've looked at this in detail say that maybe not so fast. Some are definitely biological, have microbial activity, and other ones could be abiogenic. The other part is when you actually look at these, you never tend to find any organic materials in them anyway. So they're accepted as pretty good evidence for early life, but you tend not to find a lot of, in fact, I don't know of any reports of actual sort of carbon or material coming out of these. The argument is that they're fossils and have been completely replaced. So it's not clear how useful those are either. So let's go back to the carbon isotope record. Carbon isotopes aren't, we'll get into some alternate interpretations, but the best one is that light carbon is a potential signature of life, and it's a great one, especially when our geologic record gets to be really crappy, because we only need to preserve microscopic flecks of carbon that have this isotopic signature that we can then look at and see if we think that there was life. It seems to be, broadly speaking, work, right? I've shown you this. There's the kerogens through time, the offset with the carbonates. So the idea is if you find something at 0, not life, didn't involve life. If you find something light like this, then you have life. And this fractionation actually would tell you, mostly this is introduced by photosynthesis. So here, for comparison, are C3 plants, C4 plants, so plants, here's the inorganic carbon up here. Here are some methanogenic bacteria going all the way down to -40. So these are sort of where you'd expect biogenic materials, and this is where you expect non-biogenic materials. So it's centered around minus, and we'll get into these data points here.
But how do you tell the difference between organic and inorganic carbon deposits?
We don't care what form the carbon is, right? So if you take your plant and cook it to graphite, it's not that part isn't going to change the isotopes all that much.
Right.
So the point being that when we measure it, when we look at how much 13C there is relative to 12C, if we get something like -5, probably not life. If we get something like -30, probably life.
So you're saying deposits before 3.5 billion years ago, the ratios are significantly different from what we see?
We don't have any kerogen deposits prior to 3.5 billion years. So the record actually simply runs out here. The oldest data point is at 3.5. So what I'm going to get into now is how we get an older carbon record than 3.5 billion years old. So basically, what can we find to get back into it? It's going to be, spoiler alert, these data points here. So one place, so this is work that was done, so this is, late 90s vintage, early 2000s. This is probably one of the most controversial findings that I know of in my field. And the argument is that in Achillea, southwest Greenland, there's marine sediments that are sort of 3.8 billion years old, or possibly older. We're actually going to sort of go here, and so there is a zoomed in, there's a geologic map, and the argument is that as you sort of get in here, that this is a 3.8 billion year old sort of sedimentary material. It's been cooked. This is still, we're sort of on this map down here. The argument is that you can't, unfortunately, directly date this sediment, so you have to try and date rocks that are in relation to it. And the argument is then that this is sort of 3.8 billion years or older. And there's what the rock looks like. There's a pen for scale. There's a human for scale. Kind of a messy place. And what you find are little crystals of apatite. Apatite is a calcium phosphate, and these apatites contain carbon inclusions. So that when this apatite grew, it swallowed up a little bit of graphite, and we can measure the isotopic composition of this graphite later.
Are these actually apatites or hydroxyapatites?
They'd be so apatite, the calcium. This always has either either a hydroxyl group, chlorine or fluorine attached to them.
So it's a mix. It's a mixture.
Apatites are always a mix when you find them in the crust. So They're not pure hydroxyapatites or anything. So you will have.
There's no later study. Okay, fine. Thank you.
So again, I've shown you this before in a different context. We're going to use, they used an ion microprobe, so they shot, in this case, some cesium at their sample, made negative carbon ions, accelerated them, pulled them around, and analyzed them based on mass, carbon 12, carbon 13, different mass. You can separate them from everything else. You know, that's what minerals typically tend to look like when we work on them. Those are zircons. So, the way that this was done originally is they had found these quartz, apatite, graphite, and the Moisich 1996, they did this work, and then a lot of people went, well, we don't believe you that these apatites exist, or that the graphite in the apatites exist. So this was redone by McKeegan et al. 2007, and this is the one that I'll sort of focus on, who made this really cool 3D model. So the purple here is quartz, the apatite is shown in blue, and then the graphite is this gray thing here inside of it. And this was done using a Raman system where you shine a laser at it and look at the spectroscopy, so you never actually had to expose this to the outside world. So there's no chance of actually contaminating it. The cutaway here is just to show it to you. So there's absolutely graphite. And the carbon isotope, the composition is minus 29 per mil. So that at 3.8 billion years, we have something that looks like it's really isotopically light. Not unique to light, we'll get into this complication at the end, but certainly suggestive. So the question that I want to ask is, can we extend this into the Hadean? Can we get this into the 4 billion year period? So unfortunately, so this is work that I was involved in. The carbon inclusions that I will show you are ones that I found with a Raman system. And the study was then by Beth Ann Bell. We did this in collaboration to do the carbon isotope. Fortunately, we weren't the first to take a bite of this apple. Menniken et al. in 2007 claimed that basically every zircon from the Jack Hills that they looked at had either graphite or diamond in it. Diamond is kind of hard to get in, and secondly, they polished their samples with diamond paste. Now, Larissa Dobrinetskaya asked them for their original samples, asked for permission to take a tiny, basically take a tiny little cut across here where they're claiming there's diamonds, and to analyze it with a really high resolution microscope, a transmission electron microscope. And I absolutely love this paper and love this result, because what she found is, here's the zircon. So this is now a transect through the crack. This is now 500 nanometers is this scale bar. So we're only really looking at a tiny, tiny portion of this. But here's what you find. This is zircon. There was K feldspar originally there that was exposed at the surface. K feldspar is a common mineral in granites, and it's really, really soft. So what happened in the polishing process is they preferentially dug out the K feldspar and then deposited, and then some diamonds from their diamond paste got stuck in it, and even better, they also put epoxy into it, because these were mounted in epoxy. The cooler part is this also explains the graphite finding. Graphite is what you get when you take epoxy and heat it with a ladle. So, this was a very nice reputation of actually 2 Nature papers. So, this, however, sort of inspired us because at UCLA, where I did my PhD, we had a really large collection of these old zircons, and so we could basically do statistics, right? If you've got 1,000 of them or 3,000 of them, you look and see, do I find carbon? No, right? I mean, you've got 1,000 to try, so you're not, you can find low probability things. So we wanted to figure out what is the true rate of occurrence of carbonaceous material. Is there any, and if so, what's its isotopic composition? So the way that this search was conducted is I took some of these grains and we used the Raman spectrometer. We've already shown you what some of that reconstruction looks like, but what happens is, sorry, this doesn't actually look all that great. Sample is down here. You shoot a laser at the sample, and then the molecule, each of the molecules that are in there is going to react slightly differently to this laser light, and you're going to scatter some light that is a unique fingerprint of the material that is in there. And so you basically collect that light, make a graph of it. This is what graphite looks like from work by Biogenic Life. So at a Raman shift of basically 1300 and 1600, you should see these peaks. Sorry, so here is, it's really hard to tell what you're looking at here. So this is this feature here. This is a zircon. There are 3 black dots here. And one of these, I'll show you the Raman spectrum for, is first off, we wanted to make sure that we weren't looking at epoxy. So the first thing we did is make sure our laser power was low enough that we weren't going to cook our epoxy. Once that was done, we got a Raman spectrum of the epoxy. That's in red. And what I want you to notice, this is an OH bonding. So this is basically something that's got a lot of water in it would do. The vibrations out here. And I want you to notice that it's really prominent in the epoxy and it's absent in the black inclusion. So the black inclusion has got the features that we look for in graphite. This peak is a little broad. It's probably not structurally graphite. It's probably seen a little bit of damage. But this here looks very different than the epoxy, so we're not looking at contamination. We're looking at carbon that is actually in our zircon. And the cool thing is, actually 2 out of the 3 have graphite in them. So we have 2 carbon inclusions in one 4.1 billion-year-old zircon, and that this is the oldest dated carbon from this planet that we've ever found. Subsequently, we've looked at more, and here's now pictures taken on a much better microscope. So here's a different zircon. This black stuff, all of this is carbon, fully enclosed in the zircon. Here is more carbon. This is on a crack. You can see that there's a crack here running through it, so this is probably not the best thing to analyze because this crack allows for later fluid ingress and could mess up your signature, right? If your mineral is cracked, then you have an easy path that exposes what you have to the outside world. Maybe fluids got in there, messed some stuff up. Maybe the graphite itself is younger. You have a path for this. And so we're going to focus on, first on this 4.1 billion-year-old zircon here. And then we decided, and this was the most challenging part, is that we wanted to be absolutely sure that there weren't any cracks. Finding really tiny cracks, however, is difficult. An optical microscope can't show you a feature smaller than maybe a micron or something, or maybe 500 nanometers. But if you go to a facility that can produce a lot of X-rays for you, X-rays can look at stuff with a spatial resolution of 40 nanometers. So you can look at much smaller features, and so we asked the question, A, would you be so kind as to analyze our zircon? And we found some nice people who took it out to a synchrotron for us. And then can we take a picture? So here's one of the images, and here's the carbon inclusion, and this is the other carbon inclusion, and there's a sort of a 20 micron scale bar. So we actually had to cut this out of our zircon. So for this you use a technique called a focused ion beam, where you shoot high energy gallium at the sample, and that basically cuts away the zircon. And then this is a needle that was attached to it to lift it out and hold it. So we had our little piece of zircon. But you can see that everything is still away from the surface. So we haven't actually touched our carbon inclusion yet. Everything is still tamed, and we're using the X-rays to look through it. So Crystal Chi and Wendy Mao went to the Stanford Linear Accelerator, and were so kind as to analyze this for us. And what I'm about to show you is a movie of the X-ray transmission through it, and you can see, might be a little hard to tell with the lighting in here, but there's one, there's the other inclusion. And what I want you to see is that there really aren't any cracks that are found there. So basically, you rotate this around, you can look at this from all angles. This is like, if you've ever had a CT scan done, this is the same thing. So for CT scans, you're looking at a sort of 3D reconstruction of bones for medical purposes. In our case, we wanted to do this to image this from every possible side so that we could see if there were any cracks or features hiding from us. And the cool answer is, there aren't any in there. We have subsequently shown that when there are, so now you might ask, well, Patrick, you haven't shown us a zircon with a crack in it. How do we know that this is at all sensitive? I didn't put the images in because we're still working on it, but we have actually taken zircons where we know that there are cracks and put them in, and we can see them plain as day. So the technique does actually work. This basically works on the fact that zirconium, being so heavy, is a great X-ray absorber, and anything that you would put in that would be lighter is not going to absorb them as much. So you're going to see that contrast difference, which is why the inclusions are dark. It's because carbon doesn't really absorb X-rays, and the background around here, this is just air, which doesn't absorb any X-rays, so stuff that doesn't absorb is shown as black, and this is why the inclusions are shown as this sort of darker blends in here, because they are absorbing it less than the zircon. For example, the needle is made out of, let's say this is a tungsten needle. So a very heavy thing that is a graded absorbing X-ray. So basically the color range goes from really great at absorbing, not so great at absorbing. And then we had to, and this is the most nerve-wracking procedure ever. I wish I had actually videotaped it, but the person who did us the favor of doing this process is, you then have to, you then can't do mass spectrometry on this, because you can't just stick your needle into the sample. You actually have to press it into indium metal. So you have to take the needle, lay it flat on the surface, and press it into indium and hope that the sample stays. Christopher Sneed, grad student there at the time, did a fantastic job with it. Not only did he get the sample in the indium, but he got the right side up. How he did that, I don't know.
With respect to the image that we're looking at, is that X-ray diffraction or X-ray fluorescence?
This is just transmission. So this is just
simple transmission.
This is just simple transmission. Basically, in the black, 100% of the X-rays make it through the sample, and in the white, none of them make it through the X-ray, and this is some fraction of the X-rays make it through. So you're just looking at very simple, what part of the beam got transmitted through, and the beam is larger than the sample. So you can, the way that you figure this out is you then take the sample out, take the same picture again, and see how much makes it through.
With respect to your Raman spectroscopy, are you using straight Raman or are you using an inverse system?
Nope, just straight Raman microscopy with a transmitted light microscope. You can see, right, the side that we wanted is where they're closest, so it's sort of this side here. This is the side, right, that we wanted. If we had got one of the other ones, it would have been really hard to go in and find it, so we actually, you know, kind of wanted this surface up. Amazingly, that actually happened, so we could do this entire project. The sample that had flown around the country, been in many analytical techniques at that point. The entire analysis was resting on our ability to press it to metal. Luckily, that all worked out.
Can you tell us how big that sample was?
This is, the previous image had a scale bar, so this, you're looking at, you know, there's a scale bar, 20 microns, so this is maybe 60 microns across or something.
So a human hair is 100 microns?
Yeah.
Okay.
So you're looking at something smaller than a human hair on the needle, and the needle itself, annoyingly enough, is flexible. This was incredibly nerve-wracking and a giant sigh of relief when we actually got data from it.
I hope you bought that guy a beer after.
A couple. So here's now, this is now age, post-mineral age, carbon isotopic composition, and here are the previous ones, right? This is the Achillea, this is the Greenland stuff that I showed you previously in the apatites, plotted sort of here at about 3.8, and then here's our new data. Both of them gave very similar results, minus 25, right on where you would expect organic carbon, and well resolved from inorganic. So this could be the earliest evidence for life on Earth. The caveat to this is that there are some other processes that could possibly do this. Meteorites, in fact, have carbon isotopy. They're minus 12 plus or minus 6, so if we plot them, they would sort of, forget the age, just look at the vertical, right? It's possible that the tail end of the carbonaceous chondrite range hit this, and that we basically are seeing meteoritic carbon that was subducted and cooked. So that's a possibility. People have argued there's a mineral siderite that can, it's an iron carbonate, this can break down and give graphite, but this only shifts you by minus 6 per mil, so that would get you about to minus 11, so that's not enough. There is an entire class of synthesis, Fischer-Tropsch synthesis. We don't need to really get into the details. This is possible of fraction. This is capable of fractionating carbon isotopes, but evidence for its operation on Earth. It's great to make this stuff go in a lab, but it's not actually found. Evidence for it is not actually really found on this planet. The people who do this sort of have to admit to this. It's possible, but we don't see any evidence that this occurred. And then I think the simplest and most likely explanation is that this is biogenic carbon, but there's ways to test this going forward in that, right, if it is carbonaceous chondrites, we would expect some distribution that ranges all over this place, and if we find more carbon inclusions and they're sort of centered here and have a similar, then maybe we're just seeing that. But if we do this and we find a whole bunch that are down here, then this starts to look like a more compelling case. So as we go forward, it takes a while to get each sample sort of through the pipeline. You've got to find the carbon, you've got to dig it, you've got to cut it out, you've got to take it to a synchrotron, do the X-ray imaging, then you've got to mount it, do the mass spectrometry. But so we're hoping to get more samples sort of process. This was published in 2015. I started working on this in somewhere around 2012 is when the first, so it took us about 3 years from finding the first one to actually getting the paper out the door. So that sort of takes us nicely to the conclusions. I think the oldest fossils are 3.4 billion years. The oldest evidence for life, potential evidence for life maybe, is 4.1 billion years old based on carbon isotopes. We're not sure how life itself originated. The environment, I would argue, as we've gone through in all of the previous lectures, was probably fairly similar to the present day. You're looking at an atmosphere with carbon dioxide, water, not the sort of methane stuff that's been used in analog experiments, and that this is a topic where the sole amount of data, the sole amount of carbon we know to be older than 4 billion years is represented as these two data points and is now coding the inside of a mass spectrometer. So we don't have really a great record going back, but it seems that the harder and harder we look, the more we find it early on. People have now, this age was highly controversial in the late 90s, this sort of 3.8 billion year old origin of life, and now people are, sort of routinely writing papers arguing, I didn't get into them so much, but people sort of now accept it back to about 3.9. So there's skepticism of our work here, but I hope that future work will prove us right. And we'll hope to do some of this work. And I'm just going to show you next time. We're going to talk about impacts and meteorite strikes and what that effect would have. What we know about them, what the bombardment rates were. There's something called the late heavy bombardment at 3.9 billion years, where there's the argument that Jupiter moved around in its Venus. We'll take a look at that. Jupiter moved around in its orbit, and we'll take a look at that and see what we know about the early impact record and how destructive a place the early Earth was. So thank you, and I'll take questions.
We'll have to talk later, but are you familiar with the Fischer-Tropsch processes?
A little bit.
Oh, too bad. All right. I'll talk to you later.
Okay, in the back.
So we would use carbon deposits in meteorites as a benchmark for carbon isotope ratios, right? We need something to tell us this is what it should look like if there was no life here. So we would look for carbon in
if you look at
rocks from the moon or from meteorites, right?
The best thing that we look at is the mantle. So rocks from the mantle, and they are isotopically, this is the inorganic carbon field here. So rocks from the mantle are -5 Vermel. So that gives us the benchmark of what it should be. If there's not
those rocks from the mantle from volcanic deposits.
Like you can look at like mid-ocean ridges or oceanograms. You can also look at volcanic ones, although those tend to be contaminated by stuff being subducted down. We know we're subducting organic carbon. So there's a little bit of a spread there, but it's always in other isotopic systems associated with subduction of organic carbon. But the benchmark is basically we should be finding something in that inorganic carbon range. That's established not necessarily from the meteorite record because we don't know how representative that is of the Earth. So we benchmark it to the Earth itself.
As you know, I was going to ask you a question after the lecture. You took care of those analytical questions in spectacular fashion.
Thank you. Other questions?
Yeah, I guess it's interesting when plate tectonics started. Could have been carbon life earlier, but it's been subducted deep into the Earth's mantle.
It's entirely possible that we will never know when life originated on Earth. So there's a couple of things. A, you rightly pointed out, subduction could have destroyed the evidence for it. We might not see it. We got incredibly lucky to have any carbon from that time period at all, I would argue. This is a sort of 1 in 1,000 Hadean zircons percent. There's about a 1 in 100,000 probability that the zircon from the Jack Hills would be Hadean and have carbon in it, so this is not a trivial undertaking. To find that data point is years of work of simply finding enough zircons at all so that you can conduct such a search. The other thing is that fractionating carbon isotopes tends to happen by already biologically advanced processes. So you could entirely say that the origin of life maybe did not fractionate that life early on, and that it had to get to a certain point, right? If you look at the C3 and C4 plants, This is happening in photosynthesis. So basically, we are more sensitive to certain parts of the biologic arena than other parts. And there's forms of life that do not necessarily fractionate carbon. So it's possible that our earliest record, we're only able to see it, even if we have this perfect record, and we can all agree on the measurements and everything, that we might not actually see this origin because it might be invisible to us, either through subduction or because it's been cooked to heck and it didn't fractionate the carbon isotopes in the first place. In the back?
Thank you. We don't know when modern oxygen concentrations emerged in the atmosphere. Do we have any kind of benchmark, any data at all as to
the oxygen became prevalent in the atmosphere about 2.3 billion years ago. So about 1.5 billion years more recent than this. So basically, there was very little evidence. There's very low oxygen through this time period. People argue at about 3 billion years, which is sort of off the scale here, that there was a little bit of oxygen. Then, at the great oxidation event, is when it really became prevalent about 2.4 billion years ago.
Why don't we see more complex life emerging much earlier?
That's an excellent question. I have absolutely no idea. The only speculation I'll give you is that we can see diversity explode in recent times, and this is entirely speculation on my part that I may not even be qualified to conduct, is that maybe it was simply much harder to get something working that could survive. If your life is really primitive, say your cells aren't able to filter the ions that are coming in, they haven't developed those membranes quite yet, maybe they're not as robust, and so there's a lot higher failure rate to mutations than at recent times when life has gotten more established. So maybe it just was that early part of the process was a lot harder than the later part of the process. But I really don't know, and that is a really interesting question, independent of when the origin of life is that most of the evidence, most of the history has been wrapped up in this very simple life, and then at some point it exploded. We don't, particularly with the Cambrian explosion 500 million years ago, where there's not necessarily a great explanation for why that has happened. Yes?
What is the earliest evidence for cyanobacteria? Back how far?
So the earliest fossils are argued to be cyanobacteria, and so that would be 3.4 billion years. So those would be argued to be cyanobacteria.
The problem with cyanobacteria is they're highly integrated cell structures already.
Yeah.
Right. So you've already had the formation of the lipid bilayer.
Yeah.
Which would be the capsule. Okay.
Yes.
The problem is you've also got chloroplasts, which are again organelles of a highly integrated structure, and using the argument that these are absorbed, and you go on to eukaryotic organisms. You're really not talking about anything that has occurred through processes not associated with cellular life in existence. So
we don't know what the process is that gave us life, right?
and the materials that you're looking at with respect to the zircons have already been metamorphosed to quite an extent. The oldest rocks are heavily metamorphosed. The zircons formed in magmas, so there's not pleasant environments for preserving this stuff. So it is not. The arguments for them being cyanobacteria are morphological arguments, in that it looks like cyanobacteria does, and then there's some. And so this is a work that's done by a modern analog, because unfortunately we can't pick up anything much older. So there's fairly severe limitations about what we are able to say about this time period, and so we can basically look for a little hopeful glimpse that there was some carbon whose isotopic composition is consistent with life, but is certainly not completely unique in that direction. Other questions?
Yeah, I was wondering about the complexity problem. Is there an enormously complex jump going from an RNA to a double helix?
I would think so, but I would recommend you ask a biochemist. Other?
Yes. I've read that recently many people think that the original life was a mess. In other words, if you look at bacteria, archaea, and eukaryota, they have mixes of stuff inside them that look like the original system was one with a lot of lateral gene transfer, viruses, plasmids, and stuff like that. So the original life might have been a pretty messy soup.
Agreed. That is entirely possible. There are some ideas. I found some of them. I didn't list them on my slide of possible origins of life and features that we know. Right? Everything, the further back, when you look at the further down you look at the sort of the tree of life, right, the argument is everything goes back to one ancestor and has had to split off from there. So, or at least the life that survived at the present day, we don't know if it started more than once or not, for example. So you would expect a lot of intermixing, especially in the early time period, right? Especially if everything sort of goes back to one place has been argued. Other questions?
Just one simple thing just to clarify in my mind.
Sure.
Do we think that all life today is descended from one, one organism, one first life? Or are we descended from many?
The argument is that it goes back to one, that there was a last universal common ancestor. But that doesn't mean that all life, right, that can't then be used to say that it only happened once, because the other evidence could have simply died out over time, and we might not find it. We find a chunk of graphite, and we certainly can't get its genome or anything because all that's been destroyed. So that is a much harder question to answer. Is if it only started once, if it started and failed, if there were different ones and then one won out in the end. Your guess is as good as mine. All right. Thank you very much.